Pathogens that have evolved to escape recognition by phagocytic receptors are taken up by tissue dendritic cells through the process of macropinocytosis and can then be presented to T cells. This is thought to occur after intracellular degradation of the pathogen to reveal components that trigger activation of the dendritic cell. Bacterial DNA containing unmethylated CpG dinucleotide motifs induces a rapid activation of dendritic cells. This probably occurs after recognition of the DNA by an intracellular receptor called TLR9. Exposure to bacterial DNA activates NF-kappa B and mitogen-activated protein kinase, MAP kinase, signaling pathways, leading to the production of cytokines such as interleukin-6, interleukin-12, interleukin-18, and interferon alpha and interferon gamma. In turn, these induce and augment the expression of co-stimulatory molecules. Bacterial heat shock proteins are another internal bacterial constituent that can activate the antigen-presenting function of dendritic cells. Some viruses are thought to be recognized inside the dendritic cell as a consequence of the production of double-stranded RNA in the course of their replication. Viral infection also induces the production of interferon alpha by infected cells. Interferon alpha is one of the cytokines that can activate dendritic cells to express co-stimulatory molecules. Dendritic cells are likely to be particularly important in stimulating T cell responses to viruses, which fail to induce co-stimulatory activity in other types of antigen-presenting cell. Viruses may infect dendritic cells by binding to any of several molecules on the cell surface or after being engulfed but not destroyed by immature dendritic cells. Such viruses synthesize their proteins using the dendritic cell's own protein synthesis machinery, leading to surface expression of viral peptides by MHC class I molecules, just as in other types of infected cell. Viral peptides will also be presented on both MHC class I and MHC class II molecules as a result of uptake of viral particles by phagocytic receptors such as the mannose receptor, which can recognize many viruses, or through macropinocytosis. The mechanism by which peptides generated by degradation of viral proteins in the endosomal pathway can be presented by MHC class I molecules is not known, nor, in fact, whether there is only one such mechanism. Nevertheless, it is clear that the extracellular proteins taken up by dendritic cells can give rise to peptides presented by MHC class I molecules. In this way, viruses that are not able to infect dendritic cells are still able to stimulate effective immune responses. Thus, any virus-infected cell is able to activate naive CD8 T cells, generating cytotoxic CD8 effector T cells that can kill infected cells, and also to activate CD4 T cells that can stimulate the production of antibodies.